I will start in 2-3 minutes. Right, so I'll share my slides. So this is the 12th and final live session of this course, Structural Biology. As always, I will be covering the lectures of week 12 and end with some practice problems. And the slides and the video will be uploaded. So even if you're not attending the class, you can check it later. So this class is about drug discovery. Drug discovery is the process by which new candidate medications are discovered. Basically, small changes in the chemistry of a molecule can greatly affect its biological activity. As you know, in the basic building block of DNA is the nucleotide, which consists of a 5-membered sugar or pentose sugar, a nitrogenous base, which can be of 2 types, adenine, thymine, cytosine, and guanine, and a phosphate group. So this nucleotide is the basic building block of DNA. And we also know that in DNA, there is something called complementary base pairing, where adenine will always pair with thymine by 2 hydrogen bonds, and cytosine will always pair with guanine by 3 hydrogen bonds. Now, in drug discovery, the basic steps are, firstly, we select a target. We select either a protein that we want to target or DNA that we want to target. The next step is lead discovery. where we have an initial molecule which shows some basic level of binding with the target. Then, using medicinal chemistry, that is from known biologically active compounds or from commonly known active compounds that have certain properties, the lead molecule is optimized. So a lead molecule which initially shows like micromolar level of binding is optimized by medicinal chemistry to get higher binding like nanomolar level of binding. Then in vitro studies are done, that is inside us, inside the cell, and in vivo studies, that is in a living organism, till finally the drug molecule is put out. And of course, there are human trials that are done at the final stage. Now, just an example of this thing, that small changes in chemistry can make great changes in biology. An example of this concept are the nucleoside analogues, also known as Na. These are compounds with identical hydrogen bonding properties of the natural nucleosides, but when they are incorporated in a DNA chain, they block the synthesis of the DNA chain and lead to cell death. We just saw the nucleotides and nitrogenous bases in this slide. So here you can see the original nucleotide has a OH at this R position, which is forms adenosine. But when the OH is replaced by a H, it is called 2' deoxyadenosine, because at this 2' position, the oxygen is lost, and instead of OH, there is a H. This molecule, 2' deoxyadenosine, is a nucleoside analog, which will behave similar to a nucleoside in terms of hydrogen bonding, because you can see in hydrogen bonding, the sugar is not involved. It is just the nitrogenous base. So because the nitrogenous base is unaffected, it will have similar hydrogen bonding properties to the natural adenosine. But when it is incorporated in a DNA chain, because here instead of OH, you have H, the chain will not be able to propagate. You can see that the OH is very important to form phosphodiester bonds for propagation of the DNA chain. But when it is 2' deoxyadenosine, the chain propagation cannot happen and the chain terminates, and this leads to cell death. Similarly, instead of guanosine, you can have 2' deoxyguanosine, where again here the R, which was OH, is replaced by H. And similarly for cytidine and thymidine. These are called nucleoside analogues, and they can work as a drug. And this is a very classic example where a small change in chemistry, just replacing an OH group by a H group, is leading to a great change in biology, that is cell death. So there can be other kinds of nucleoside analogues also. One we showed here is where the OH is replaced by H. Another is where you can see that has been some other changes in the sugar moiety, like instead of a 5-membered ring, this is a open chain sugar molecule. Sometimes sulfur can be incorporated and so on. So different changes like this lead to different effects. And this is an example how a small change in chemistry can greatly affect the biology. Now we will talk about lead molecules. A lead molecule is a small molecule that serves as the starting point for an optimization involving many. In drug discovery, a lead molecule is a small molecule that serves as the starting point for an optimization involving many small molecules that are closely related in structure to the lead compound. So basically, we can have a wide range of molecules having similar structure and similar functions, but they can show different levels of activity. You can see here an activity chart is shown where the blue corresponds to low levels of activity and the red corresponds to high levels of activity. So you can have, so this is the basic skeleton of the molecule. Now, in different positions on the phenyl rings, different groups can be substituted, and that will affect the activity in different ways. A lead compound is the compound that we start with as our initial drug candidate. So many organizations maintain databases of chemical compounds. Some of these are publicly accessible and others are proprietary databases, meaning that they can only be used inside the organization. Now, in all this database of chemical compounds, for the specific function you want, there can be a specific compound which shows a low level activity somewhere around, you can say, 10 micromolar. Now, that is the hit. Now, from this, it is optimized and narrowed down by different tests to reach the lead molecule, which shows much higher activity compared to the hit molecule. So if the hit molecule shows an activity of around 10 micromolar, the lead molecule will show activity around 10 nanomolar, which is much higher binding constant. So this lead molecule is the starting point for the optimization, and this is further optimized to get a higher binding drug, which will eventually become the drug that is used in medicines. So basically, how this lead compound is obtained. Firstly, there can be a list of conventional marketed products. So the conventional drugs that we use, like paracetamol and others, those are common molecules which have biological activity. Another can be the biologically active molecules that come from plants and different herbs. So examination of biologically active compounds found in plants. So all these molecules form a database, and first we have to document the properties of these biologically active molecules. Then from these molecules, we have to carry out in vitro and in vivo experiments to find out which of these molecules have the specific activity that we want. So if sometimes the molecule may need to be chemically modified to make it less toxic, because a drug definitely has to be tolerated by the human body. So sometimes some chemical modifications are needed to improve the binding of the drug or to make it less toxic or more efficient. And that will give you the biologically agile compounds, which are more effective drugs. Sometimes combinatorial chemistry is also used. So what is that? Suppose you have one molecule which has a property of reducing fever and another molecule which has a property of increasing immunity. So you can combine these two molecules to get a combinatorial molecule which has the property of both these molecules. So sometimes, depending on what function we want, combinatorial chemistry is used to build the initial lead molecule. After that, the toxicological and pharmacokinetic parameters are studied through application of drug delivery methods. So what this means, toxicology is checking whether it is toxic to any part of the human body. And pharmacokinetic means that we are studying how the drug is being metabolized, how it is being digested and absorbed in different regions of the human body. After that, eventually, it goes to clinical trial exams where it is tested on humans to find out the efficacy of the drug in human body. So this is a huge process which takes several years. We'll start with the traditional drug discovery methods. The traditional drug discovery involves the origin of drug discovery that evolved in natural sources, accidental events, etc. It was not target based and it was not systemic or logical like it is now. So one of the most earliest traditional drug discovery methods involved random screening. This involved collection of synthetic compounds or chemicals or natural products, which could be assayed by experimental assay method. And it involved two major approaches. One is a selected class of compounds like alkaloids, flavonoids, or other synthetic library is screened to check their activity. So you can see there are around 500 to 1,000 compounds from medicinal plants, which are screened to find their biological activity, and this process takes 3 to 5 years. The two ways of doing is that One is screening a particular class of compounds. Like you have a class of compounds called alkaloids, so you screen all the alkaloids. The other method is randomly screening traditionally identified medicinal plants from Ayurveda. So you know that from tradition and history that certain plants have medicinal properties. So you randomly screen the medicinal plants. This entire screening process of 500 to 10,000 compounds can take up to 3 to 5 years. From that, it is narrowed down to around 250 compounds, which show some level of activity. Then the preclinical trials are carried on out in vitro as well as in vivo to study different characteristics like toxicity and efficiency and so on. This takes 1 to 2 years. And these 250 compounds are narrowed down to 5 compounds for clinical trials. Clinical trials are the trials on human, and that takes a long time because, as you know, lifespan of human is very long. So if the clinical trials have to be done properly in a large enough sample size, it takes 6 to 7 years. And finally, these 5 compounds are narrowed down to one new drug, which needs to be approved by FDA. So this entire process takes multiple years to get through. And this is the random screening process where you just start with a certain class of compounds, or you start with some traditionally identified medicinal plants from Ayurveda. The other thing is the trial and error method, which includes berries, roots, leaves, and barks that can be used for medicinal purposes. So here what you do is that from the plant itself, you isolate the compound and characterize the compound by different spectroscopic methods. And then this compound is put through this process again. Now, this is a trial and error method because this is for the plants for which the activity is not known. So random screening is for the traditionally identified medicinal plants. But suppose you have an unknown plant for which you do not know the medical purpose. For that, this trial and error method is used. The biologically active compound is isolated from the plant and then tested for that it shows biological activity for which purpose. For example, cinchona bark contains quinine, which was used to treat fever in malaria traditionally. And neem leaves can be used as anti-inflammatory or antibacterial properties. So how these were found? These were totally found by trial and error method. These are all traditional drug discovery methods which have now gone out of use. Another is the ethnopharmacology approach. So this is the study of medicinal plants used in specific cultural groups. So you have native people and researchers observe how the native people treat different diseases. So they write down their observations and experimentally investigate the drugs that native people are using. And from that, they have discovered several possible drug candidates. For example, Andrographis paniculata, this is a plant. It was used for dysentery in ethnomedicine. So traditionally, tribal people used to use this as medicine to treat for dysentery. From this plant, compounds have been isolated and it has been found that which particular compound in this plant is responsible for the anti-dysentery activity. So ethnopharmacological approach involves observing native tribal people and seeing which plants they use to treat different diseases and isolating the drug from those medicinal plants. The next approach is the classical pharmacology approach, which is a function-based approach. So this is done without prior identification of the drug target. Anciently, drug discovery processes were often based on measuring a complex response in vivo, such as prevention of experimentally induced seizure, lowering of blood sugar, and so on. So what they had seen is that when that particular plant or compound was ingested, it prevented some disease like seizure or it lowered the blood sugar. So This process started by looking at the function of the compound and then identifying the structure of the compound. That is why it was called the function-based approach. In this approach, they did not start by saying that we want to target this DNA or this protein. Instead, they started based on function. They saw that a particular plant is responsible for a certain function, and then they found out which compound in that plant is the one which has the function. For example, discovery of foxglove, this is a plant in Europe, which was used for drug preparations, and it was found that this compound, cardiac glycosides in foxglove, which is extracted from this plant, has been used to prevent different cardiac diseases. So the classical pharmacology approach looks at the function of a compound and then traces back to which chemical compound is responsible for that function. And definitely one of the traditional drug discovery method is by serendipitous discovery. That is, by luck or by chance, a drug has been identified. For example, in the 1670s, bacteria were identified by Van Leeuwenhoek, and the relationship between bacteria and diseases were gradually set up in the 19th century. From that time on, researchers have been trying to find better and better antibacterial agents. So the discovery of penicillin was completely by luck, and that is why it's called serendipitous discovery. In World War I, around 18% death caused by wound-related pneumonia was reduced to less than 1% by penicillin. And such an incredible drug was discovered completely by accident when it was observed suddenly that wherever there was penicillium, the bacterial drug growth was inhibited. So this discovery was completely by accident, and sometimes drugs are discovered that way, which is called serendipitous discovery. Now, there are several challenges in the traditional drug discovery methods. The first is the process of bringing a potent drug to market needs a huge investment in time and money. Almost 70% of the funds are wasted on account of failure to pass through clinical trials due to a lack of efficacy or adverse side effects. So you can see clinical trials here, most drugs stop at this stage. They don't pass this stage because either they are not that efficient or they are toxic to the human body. So 70% of the funds are wasted in this stage. And another thing is that the time and cost enhancement to push it through the clinical trial stage leads to increase in cost of the drugs. But the problem is that there are several drugs in the market now, and because of increase in cost of the drugs, the common consumers are not able to afford it. So that is also a problem in this drug discovery process, that when so much time and money is needed, the cost of the drug becomes higher than possible for the common man to afford. So that also has to be brought down. Now we will look at rational drug discovery. There are two main classes of rational drug discovery. One is the ligand-based drug discovery, another is the structure-based drug discovery. So in ligand-based drug discovery, the 3D structure of the biological target is unknown. And what we have is we have a set of geometric rules and or physical chemical properties obtained by QSAR studies. So I'll go through this one by one. What this means is that we don't know the biological target, what DNA or protein the drug is supposed to target, we don't know that. What we know is that we want the drug to have certain physical and chemical properties. We have a library of compounds which are known to be biologically active. QSAR stands for Quantitative Structure Activity Relationship. So this structure activity relationship gives us an idea that what kind of structure will lead to what kind of biological activity. So based on that, we will design the pharmacophore. Pharmacophore is the ligand molecule that we are designing. So the design will be based on the structure activity relationship. So depending on the activity we want, we will design the structure in that way. So the physical chemical properties of the drug that we want are fulfilled. So like that, we design the ligand and test the ligand either computationally by docking or in vitro or in vivo studies to find out the efficacy of the ligand. That is the ligand-based drug discovery, where the drug is being discovered by designing the ligand. The biological target is unknown. The other method is the structure-based drug discovery, which involves molecular interaction calculation between each molecule to be tested and the biological target, which is usually a protein. So in structure-based drug discovery, the biological target is known, and we have a list of possible drug molecules that we are testing with the biological target. And we are checking the binding affinity of the ligand for the target. To evaluate this affinity, often a scoring function is used. We'll learn about this later, which gives us an idea about which molecules are high binding. So for this method, the 3D structure of the target must be known because we are checking the binding affinity of the ligand with the target. That's why it's called structure-based drug discovery, because we must know the 3D structure of the target. Now we look at a rational drug discovery example for the target cimetidine. Rational drug discovery and the drug molecule is cimetidine, also known as Tagamet. So the problem was that histamine triggers a release of stomach acid. So the need was that we need a histamine antagonist which prevents the release of stomach acid. So that is the biological target. So this is the natural histamine molecule. So we want a molecule which is similar to histamine, but which has higher binding affinity compared to histamine. So it will bind to the receptors of histamine and it will block the activity of histamine. So that is what the target was, is that the histamine molecule was binding to the receptor and releasing stomach acid. If we can have a drug molecule with similar backbone as histamine, but higher binding affinity compared to histamine, then the receptor will prefer to bind to the drug molecule and the receptor will get blocked and it will prevent the release of stomach acid. So starting from histamine, analogs were synthesized with systemically varied chemical modifications. So initially, N-alpha guanyl histamine was synthesized from histamine, where in the NH2 of histamine, a guanidine group was added. So this was the first chemical modification that was done. And similarly, several other analogs of histamine were synthesized. So this was the lead molecule, which is going to be optimized now. Chemical modifications were made in the lead molecule. This is called lead optimization. For example, instead of double bond NH2, double bond sulfur was added. That was called burimamide. Or single bond sulfur here, and in between somewhere another sulfur was added. That was called thioburimamide, and so on. Several modifications were done to the lead molecule. This was called lead optimization. And each of the so several modifications were done to the lead molecule, and each of the modified molecules were treated to see their antagonist properties. One of the molecules, metiamide, was found to be very potent and orally active. So it could be a drug that is ingested orally, but the problem was that the byproduct thiourea. So this molecule was the byproduct, thiourea, and it is toxic in clinical trials. So while this shows good antagonist activity, this is a toxic molecule. So what was done? This double bonded sulfur was replaced by this group, double bonded N, which is single bonded to which is bonded to a cyanide group. So this molecule called cimetidine was found to be effective as well as tolerated by the human body. And eventually it was replaced by a even better molecule called ranitidine or Zantac, which showed even better antagonist properties. So this was how the rational drug discovery was done. And this was a structure-based drug discovery because we know the initial structure that we are starting with. And uh, then we modified that structure into a lead molecule, and we optimize the lead molecule to get the best binding as well as confirm that it is not toxic in any way to the body, to ultimately end up with the drug ranitidine. So now we will look at ligand-based drug discovery and QSAR modeling. Quantitative structure activity relationship models or QSAR models are regression or classification models used in chemical and biological sciences and engineering. Basically, QSARs are mathematical relationships linking chemical structure with biological activity using physicochemical or other derived properties as an interface. So what this means is that from the molecular structure, you have a set of molecular descriptors. We will look into what are molecular descriptors in the upcoming slide. Just think that molecular descriptors are certain properties of the molecule, like bond angles or hydrophilicity or lipophilicity and so on. So these molecular properties are referred to as molecular descriptors X. And these so these are chemical properties, and based on these properties the molecule has a function that is being called as the response variable Y. So Y equal to Fx means the molecule has a certain function or activity based on X, that is based on its list of molecular descriptors. So that is what structure activity relationship is, that based on the molecular structure, that is X, you have a certain activity that is Y. So how this is generally found? How is this structure activity relationship generally found? Typically, we use a number of chemical compounds with known values of the activity. For each chemical compound, we calculate a series of parameters called chemical descriptor, that is X. So then we do a line fitting. We on the y-axis, these are all. So we have a set of known compounds for which we know the function, that is Y, and we know the molecular descriptor, that is X. So we plot this in a graph and we carry out a linear regression. So this will give an idea about, so now from this linear regression, you will have the function, that is you will be able to predict from a list of known compounds, you will be able to predict the structure activity relationship once you get this function. So basically, how this function is validated is that the final step is to check this function or algorithm to see if it can predict the property values for other drugs. This is called the validation of the QSAR model. For example, so from the known compounds, we are creating a function which will predict the relationship between function of the molecule and structure of the molecule. So now if we have an unknown molecule for which we will check if the unknown molecule is fitting this equation. That is, if we put the structure of the unknown molecule and the function of the unknown molecule, whether it will fit this equation. And if it fits it, that is, then we can say the QSAR model is validated. So not only, so we have to check it against several unknown compounds to confirm whether the QSAR model is a valid model. Now there are several simple rules. So from this molecular descriptors, now we'll go into more detail of what these molecular descriptors are. They basically describe some aspect of the molecule, like its structure or its property. You can have zero-dimensional descriptors, which include the molecular mass, the number of atoms, atomic mass, and so on. You can have one-dimensional descriptors, which basically say that what are the different groups present. For example, you can have a carboxylic acid group, you can have a ester group, and so on. one-dimensional descriptors define what are the different fragments in the molecule. The two-dimensional descriptors use the atom and connection information. So it will tell you the connectivity of different atoms. The three-dimensional descriptors uses 3D coordinate information about each molecule. So for each atom, you will have XYZ coordinates. Those are your three-dimensional descriptors or spatial orientation. External three-dimensional descriptors can also use 3D coordinate. So you can either have the 3D coordinates in an internal frame of reference or an external frame of reference where the molecule is docked into a receptor. And finally, the four-dimensional descriptors talk all about the different intermolecular interactions. And there can be some other kinds of descriptors like, so these are different names for the descriptors. The constitutional or information descriptors tell you about the molecule weight, number of elements, number of hydrogen bonds, double bonds, single bonds, etc. So they have information about the molecule constitution. The physicochemical descriptors tells you about the lipophilicity of the molecule and the polarizability of the molecule. So it talks about the different physical and chemical properties. The topological descriptors talk about atomic branching. And there can be other descriptors like electronic, geometric, quantum chemical descriptors, and so on. So all these are atomic molecular descriptors. That is, they form this list of X. That is the description of the structure of the molecule. Now, there are certain assumptions in QSAR. So we'll look at the assumptions one by one. One is that the effect is produced by the model compound and not by its metabolites. So we start with a lead compound. One of the assumptions we have is that the biological activity is due to the lead compound and not the metabolites. Then the second assumption is the proposed conformation is the bioactive one. So in your lead compound, you can have different processes like protonation or tautomerization, and it can occupy different conformations. So one of the assumptions is that the initial molecule, which we considered as the lead, is the bioactive conformation. The third assumption is that the binding site is same for all model compounds. So in the protein or DNA, the receptor site is the same no matter what, which of the drug candidates you are trying out. Then the fourth assumption is that the bioactivity explains the direct interaction of molecule and target. So the direct interaction of the drug with the receptor is causing the bioactivity. And finally, pharmacokinetics aspects, solvent effects, diffusion, transport, these are not under consideration yet. So these come much later in the drug development process, and we will talk about pharmacokinetics and all, but in the initial QSAR modeling, these are not considered. So we looked at topological descriptors in the previous slide, which talk about atomic branching. So there is a certain term called Wiener index. Wiener index is defined as the sum of length of the shortest path between all pairs of vertices, which represent non-hydrogen atoms in the molecule. So suppose you have a certain molecule made up of 3 atoms, say 1, 2, and 3. So Wiener index is the sum of length of the shortest path between all pairs of atoms. So between A and B, this is the shortest path. This is the shortest path between B and C. And between A and C, this is the shortest. So you add up all the shortest path, and that is what forms Wiener index. And we generally consider non-hydrogen atoms only. Here you can see a plot of alkanes where Wiener index is on the x-axis and boiling point is on the y-axis. You can see that as the length of alkanes increase, the Wiener index will also increase, because when you have more number of atoms, the sum of the paths between the atoms is larger. So for larger alkanes, that is with greater Wiener index, you can see, suppose I look at this alkane, which has a high Wiener index, it will also have a high boiling point, because larger alkanes have higher boiling points due to greater van der Waals interaction and so on. Whereas if we have a alkane with a small Wiener index, the boiling point is also low because the number of atoms is low. So this is basically, we are defining one of the topological descriptors of the molecule, that is the Wiener index. Now we look at three-dimensional QSAR. The hypothetical receptor surface model is constructed from the training set molecules three-dimensional shape and activity data. So we said that the initial QSAR function is derived from a list of known compounds. So we have a list of known compounds for which we know the molecular descriptors and we know the biological activity. So we plot these known compounds with the biological activity on the y-axis and the descriptor on the x-axis to get the structure activity relationship. That is what this is talking about, that from the known molecules three-dimensional shape and activity, we can model a hypothetical receptor surface model. And the best model can be derived by optimizing different parameters like atomic partial charges, surface fitting, and so on. And also descriptors like van der Waals energy, electrostatic energy, non-bonded energy between the drug molecule and the receptor surface, that also can be optimized. So this is basically, we get an idea of the three-dimensional surface of the receptor and the drug molecule. And that can be optimized by optimizing the different interaction energies and also the structural parameters. There are certain advantages of predicting biological activity with QSAR modeling. These are, it is able to predict the activities of a large number of compounds with little to no prior experimental data on activity. So that is what this was. Once you get the structure activity relationship, for any unknown molecule, if you know the structure, the activity can be predicted. So that is the advantage of QSAR, is that you don't have to do any experimental data. Once you have the relationship, if you know the structure of an unknown compound, the activity can be predicted. QSAR can also reveal which molecular properties may be worth investigating further. So from the structure activity relationship, you will have an idea that which are the structural features of the molecule that are important for the function. QSAR has also been regarded as a green chemistry approach, since chemical waste is not generated. You are performing in silico prediction, meaning computational predictions, and so you are not doing wasteful experiments, and so it is regarded as green chemistry. And also the in vivo and in vitro experimentation can be reduced. So these are very time consuming and expensive procedures. And if you have to do it for all possible drug candidates randomly, it is too expensive and time consuming. Rather, based on QSAR modeling, if you reduce the number of drug candidates, then it saves a lot of time and money. However, there are certain disadvantages of predicting the biological activity with QSAR. One is that it does not provide an in-depth insight on the mechanism of biological action. And another is that sometimes the predictions are very inaccurate because you are predicting it based on previously known compounds. So if the unknown compound has a new property, suddenly it will not be able to predict that. Now we will look at ligand-based drug discovery. So a pharmacophore is an abstract description of molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. For example, if you know the binding site of some protein, like suppose it has a OH like this, an OH like this, and a double bonded O. You can imagine a ligand binding to this receptor site, which will suppose it is binding by hydrogen bonding. So the OH is a hydrogen bond donor. So you must have a hydrogen bond acceptor in the ligand like a double bonded O. And double bond O is a hydrogen bond acceptor. So you must have a hydrogen bond donor like OH to bind to the receptor site. So if you can identify the binding site, based on the structure of the binding site, the ligand can be predicted that what functional groups the ligand must have. This is the pharmacophore model basically, where it explains how structurally diverse ligands can bind to a common receptor site. So any ligand, no matter how different they are structurally, if they have this kind of orientation of carbonyl and OH group, theoretically it should bind to the binding site. So using this pharmacophore model, we can identify by de novo design or virtual screening, we can identify novel ligands that will bind to the same receptor. Some pharmacophore features include hydrophobic centroids. So these are basically for hydrogen bond donors or acceptors. Suppose some part of this receptor binding site had some hydrophobic residues. Then it would prefer hydrophobic residues like methyl groups or phenyl groups, or if there was aromatic ring, there can be pi-pi stacking through phenyl groups. So basically, depending on what is there in the receptor binding site, the like pharmacophore can be designed. So if the receptor binding site has aromatic rings, pharmacophore can also have an aromatic ring which will do pi-pi stacking with the receptor and bind with it. Or if the receptor binding site has a lot of alkyl groups, then the pharmacophore can also have, you can see there's an isopropyl group here, which can have hydrophobic interactions with the receptor binding site. So basically, a well-defined pharmacophore model includes both hydrophobic volumes and hydrogen bond vectors. The features need to match different chemical groups with similar properties. So basically, you need to put different chemical groups together which match the properties of the binding sites to develop the pharmacophore. So you can see this is an example of a 3-point pharmacophore where you have a hydrogen bond acceptor, which is C double bonded O, a hydrogen bond donor, that is OH, and a hydrophobic residue, which is isopropyl here. So it is 3-point pharmacophore. Or you can have a 4-point pharmacophore where along with the hydrogen bond donor, acceptor, and hydrophobic residue, there is an additional aromatic ring which can carry out pi-pi stacking with some aromatic residue in the receptor site. So this pharmacophore concept is basically specification of the spatial arrangement of a small number of atoms or functional groups. With the pharmacophore model in hand, we can search the database for molecules that fit this spatial arrangement of functional groups, and those can be the lead candidates for the drug. So in ligand-based drug discovery, there are certain steps. First is we select a training set of ligands. That is, we choose a structurally diverse set of molecules that will be used to develop the pharmacophore model. The next step is conformation analysis. A set of low energy conformations is generated for this training set of compounds. And in this low energy conformation, the bioactive conformation is expected to be there, because definitely the conformation in which the molecule is binding has to be a low energy conformation. The third step consists of molecular superimposition, where this combination of low energy conformations of the molecules are superimposed on the receptor binding site. This combination of low energy conformations of the molecules are superimposed with each other to find the functional group that is common to all the molecules. Then we can predict that those are the functional groups that must bind to the binding site because they are the common feature of all the binding molecules. And the set of conformations that has the best fit is assumed to be the active conformation, which has the binding to the biological active site. The fourth step is abstraction, where we transform the superimposed molecules into an abstract representation. So this is the abstract representation where we are saying there must be 2 carbonyl groups and 1 OH group oriented like this. So this kind of representation is called an abstract representation. Or suppose when there is an aromatic ring, we can say there has to be an aromatic ring here. So that kind of description of the different groups and orientations is called as abstraction. And the final step is validation, where so the pharmacophore model is a hypothesis which is predicting that any molecule which has this kind of orientation of functional groups will show biological activity. So the model is only valid when suppose it works for unknown molecules also. So you have an unknown molecule which has the pharmacophore orientation, and if that molecule is found to show good binding, that means the pharmacophore model is a valid model because it is successful for an unknown ligand also. So this is the process of model development for the ligand-based drug discovery. Now we will go to structure-based drug discovery. In structure-based drug discovery, we use three-dimensional protein structure information in the development of new biologically active molecules. De novo basically means starting from scratch, that is from the beginning. And it is a process in which the three-dimensional structure of the biological macromolecule, usually protein, is used to design new targets. So you have the protein molecule which has the receptor. The structural, first step is the structural determination of the lead target complexes and lead modification using molecular modeling tools. So you have the receptor ligand complex, and the first thing to do is to determine the three-dimensional information of this receptor ligand binding site. After that, this lead molecule or drug molecule can be modified to perfectly match with the binding site of the receptor and get a like lead optimization can be done. So once the structure of the receptor is known, compounds can be built that are complementary to the target binding site, that is the receptor binding site on the protein. And this can be done via a random combination of small molecular fragments to make the complete binding molecule. So suppose you know this is a receptor binding site which has a carbonyl here. And this is just shown in very simple method. But depending on the structure of this binding site, we can put together different molecular fragments. Like you can see an imidazole ring is here and it is attached to a benzyl moiety. Now we can see that what orientation of the benzyl moiety will perfectly fit into the binding pocket of the receptor. And by doing that, the drug can also be optimized. So the structure-based drug discovery means that we need to know the three-dimensional structure of the receptor binding site. Based on that, compounds can be built by combination of different molecular fragments to perfectly fit with the binding site. And in that way, the drug can be optimized. Now we look at the steps involved in this structure-based drug design. The first is the generation of potential primary constraints. These are the molecules which set up a framework for the desired structure with the required ligand receptor interactions. So there can be two types of potential primary constraint. One are based on receptor, that is the interaction of receptor forms the basis for the drug design. So you find out which are the binding sites on the receptor. So this is the binding pocket of protein which has phenylalanine, aspartic acid and isoleucine in the binding site. So you have this phenyl ring of phenylalanine, COO minus of the aspartic acid, and isoleucine, you have just the backbone NH of isoleucine in the binding site. So based on this binding site of the receptor, the primary pharmacophore model can be designed. And the second is ligand based. So the pharmacophore model is based on the binding. So suppose a known ligand is known which binds to this binding site. So that known ligand is used as a target for the drug we are. So the drug we are designing, the structure of the drug must be in the same framework as the known ligand that binds. So that is the generation of potential primary constraints. What this means is that we find out the basic features of the drug, and how we find that out, we can either look at the structure of the receptor binding pocket and design the drug in such a way that it will bind to the binding residues of the receptor pocket, or we look at the native ligand that binds to this binding region, and we design the drug based on the structure of that ligand. So that is called ligand based. The second step is derivation of interaction sites. So we carry out atomic resolution structure of the active site to find out which residues, either the side chain or the main chain amino group or so on, which are actually the interaction regions. The next step is building up methods. So based on the interaction region, so suppose there is the backbone NH of isoleucine, the phenyl ring of the phenylalanine, and the CO minus of the aspartic acid are the interaction sites derived in step 2. The step 3 is building up methods. So we take different fragments that will bind to these moieties. Like for the CO minus, we can have a OH group, which is hydrogen bond donor. For the phenyl ring, we can have another phenyl ring, which can show pi-pi stacking. Similarly, for the NH, which is hydrogen bond donor, we can have a hydrogen bond acceptor like C double bond O. So we fit different fragments which will bind to these interaction sites. Then we can link these fragments together. So you can see 2 fragments have been linked, and that shows quite good binding compared to only 1 fragment. Or we can grow. So you have different fragments here. You can link and grow the, by linking the different fragments together, you will get a large possible drug candidate, which will bind to all the possible binding sites. So these are methods by which the lead compound is built up by linking and growing the different fragments. And often molecular dynamics can also be used to dock certain fragments and see which are the optimized fragments that will fit into that binding site, and so on. So these are building up methods by which the lead compound is built up. Then is the assay or scoring step, in which biological assays are done to find out the activity or function of the possible drug candidate. And the steps 5 and 6 basically consist of more refining. So after we find a set of drug candidates which show decent biological activity by this step 4, that is the biological assays. Then by comparing these drug candidates, we can find out which are the common functional groups, that is, which functional groups are most responsible for the binding. So we can predict that how we can add or modify different functional groups to improve the binding further. That is basically search strategies, where we search for different strategies to further refine the binding. And then we have a set of secondary target constraints. So based on the strategies that we found, that suppose the strategy is that the compound needs to have 2 carbonyl groups. So that will become part of your secondary target constraints, where you have a set of initial drug molecules from step 4. Now you will get a set of more refined drug molecules with higher binding from step 6. So this is how the structure-based drug discovery happens. Now, virtual screening, or VS, is a computational technique used in drug discovery to search libraries of small molecules in order to identify those structures which are most likely to bind to a drug target, typically a protein receptor or enzyme. So if you remember, after the determination of binding sites, we were looking for different fragments that will bind to these regions in the interaction site. So in order to search for these fragments, one of the ways of doing it is virtual screening, which will search through huge libraries of small molecules to identify which fragments can bind to different interaction sites. So, as the definition suggests, virtual screening, basically it can screen over 10 to the power 60 compounds, and it can narrow down this number of compounds to a manageable number, like maybe 100. And those 100 compounds can be synthesized and tested. So instead of testing an unimaginably huge number of molecules, like 10 to the power 60 molecules, virtual screening will basically narrow down the number of molecules into the best possible candidates, and those can be synthesized and purchased and tested. Another way of narrowing down the possible number of drug candidates is by docking. So docking will basically, you need in docking, you have the three-dimensional structure of the protein, and you have a library of small molecules. The docking software will fit the small molecule into the receptor binding pocket and give you an prediction of the binding energy. So by this docking, we, instead of a huge number of compounds in the library, we can narrow down the compounds to the best binding drug candidates. So after the prediction of the best binding molecules, only those molecules can be synthesized. There is no need to synthesize the rest of the molecules, which docking is predicting will not show good binding. So docking basically fits the molecule into the binding pocket and predicts the binding energy and the orientation of the molecule, which is the best binding. So docking predicts that in order to reduce the experiments you have to do. Now we'll talk about the molecules available for screening. So basically, real molecule, there can be 1 to 2 million real molecules, and these are basically kept in in-house archives of large pharmaceutical and agrochemical companies. And commercially available molecules can also go up to 3 to 4 million molecules. However, hypothetical molecules, which can be found in virtual libraries, can go up to 10 to the power 60 molecules. So these libraries are basically you screen through these libraries for any drug design process. These libraries can be divided in 4 groups. One is the general library, that is for broad interest in high throughput screening against any target. So these libraries will have those compounds which will show binding to many targets. So it is not target specific. And so they are called general libraries, and they are used for high throughput screening. The second kind are focus libraries that are aimed at a family of related targets. For example, a library designed specifically containing ligands against the chymotrypsin fold or against serine protease inhibitors. So it is a focus library aiming at a particular target. Aiming at a family of related targets. So the chymotrypsin protein, there are many kinds of chymotrypsin protein, and the focus library will aim at all the proteins in the chymotrypsin family. Targeted libraries are aimed at a single therapeutic target. For example, HIV-1 protease or beta-lactamase, which is a single protein target. So it is a much more targeted library compared to the focus library. And cherry-picked library is an exclusive library containing a set of compounds which we can ask a company to synthesize for us. So like, suppose we are targeting a completely new protein. So then we can ask a company to synthesize a library of possible ligands for that particular protein. That is called cherry-picked library, where ligands for the particular protein we are targeting can be synthesized by any pharmaceutical company. Now we will talk about scoring functions. Scoring function is an approximate mathematical method used to predict the strength of binding affinity between two molecules. Scoring functions are basically of 3 types. The first is the force field analysis that can calculate the actual molecular forces that exist in the protein ligand complex by optimizing van der Waals, electrostatic forces, and hydrophobicity to each force field. So this is a very complicated process. So you have different force fields for different kinds of interactions like van der Waals, electrostatic, hydrophobic interactions, and so on. The force field analysis will optimize each interaction in each force field through a very complicated process. So this takes a lot of time. The second scoring function is the empirical scoring function. And this is the fastest scoring function. It will optimize empirical terms such as van der Waals interaction, hydrogen bonding, electrostatic energy, and so on. But it will just it is a qualitative optimization of the different energy terms. So you can see in the empirical, you have different delta G van der Waals, delta G hydrogen bonding, delta G hydrophobic, and so on. These are just taken as a single term, which is optimized. Comparatively, in the force field optimization, each of these energies are optimized in different force fields. So that is a more accurate calculation, but because of the intensive calculation, it takes too much time. Comparatively, empirical scoring function will also optimize the energy, but it is a much more qualitative analysis where force fields are not considered. Each energy is given a single term and it is optimized. So it is a much faster scoring function. And the third kind is the knowledge-based scoring function, where experimentally determined structured geometrical data is used from databases like PDB. So it converts structural information from protein ligand complexes to predict that the unknown protein ligand complex, how much binding it will show. So using these kind of scoring functions, the binding affinity between the ligand and target is predicted to optimize a lead molecule. So there are two general models of enzyme activity. One is the lock and key model, where you have the active site of the enzyme is rigid and has a particular conformation, and the substrate is also rigid and has a fixed conformation. So the substrate will perfectly fit into the enzyme pocket to have the enzyme substrate complex. This will be converted into the enzyme product, and the product will be released. This is the lock and key model, because lock and key, meaning the lock will open only that meaning the key will open only that particular lock. So there are particular substrates which will fit into the particular binding pocket. That is the lock and key model. Another is the induced fit model, where depending on the substrate, the enzyme can adjust its shape. So the enzyme has a certain flexibility, so in which the enzyme can adjust its shape to fit the substrate, and then the products are formed. So this is a more flexible model, and the fit is induced. So depending on the shape of the substrate, the enzyme can change its shape. Now, if a protein binds to several ligands, so suppose a protein binds to several ligands. Now, if there are two terms, positive cooperativity and negative cooperativity. Positive cooperativity is when the binding affinity increases with ligand binding. So here you have a protein shown by this, and there are 4 binding regions of the protein shown by 4 squares. S is the ligand which is binding to the protein. When one S has bound to the protein, then the binding affinity of the other sites increase. When 2S have bound to the protein, the binding affinity increases further. So with the binding of the ligand S, the binding affinity of the protein for the ligand increases. This is called positive cooperativity. And the opposite effect, if it is there, that as when one S is binding to the protein, if the binding affinity decreases, shown by this purple square. So when one S is binding affinity decreases. When 2S is binding affinity decreases much more. So like that, with binding of the ligand, if the binding affinity decreases, that is called negative cooperativity. So computationally, lock and key is the simplest model to predict, because lock and key is a very rigid model. The enzyme shape is also fixed, substrate shape is also fixed. So there are very little flexibilities there, and there are only 6 degrees of freedom, which is 3 rotations and 3 translations. Comparatively, induced fit is much more difficult to model, because there can be a great number of degrees of freedom, because the protein can adjust, the enzyme can adjust its shape. That means there is more than 6 degrees of freedom. And so different algorithms may be needed to model the side movement of side chain, movement of the backbone, movement of ligand, and so on. So complex molecular dynamic simulations, they basically account for all atom nanosecond long molecular dynamic simulation of the protein target, that is the enzyme, to describe the conformational flexibility of the enzyme with rapid docking of the small molecule to the enzyme. And pictures of this docking are saved during the molecular dynamics run. But the problem is that often the conformational change needed to uncover the binding pocket would not be observed at the 1 millisecond time scale accessible to explicit solvent MD simulations. So basically, molecular dynamic simulations have a certain time scale of around 1 millisecond. But the conformational change that is happening in the enzyme, which is allowing the substrate to fit into the enzyme, can be of a completely different time scale. So then the molecular dynamics induced fit needs to be adjusted in such a way so that it is of the same time scale as the change in conformation of the enzyme, and only then we can study how the enzyme conformation is changing to fit the substrate. So that way it is induced fit is much more difficult, because conventional molecular dynamics, the time scale will not be able to show the conformational change in the enzyme, and much more specialized molecular dynamic simulation is needed. So what are we looking for in a good drug candidate? The first point is we want good activity and selectivity on the right target, and it should show the desired biological response. But another thing that has to be considered is the absorption, distribution, metabolism, and excretion. So it must be properly absorbed and distributed to the site of action. The drug must be metabolized. It cannot accumulate in the body. It must be metabolized and excreted. So this is ADME, absorption, distribution, metabolism, excretion. And also we must keep in mind the toxicity. It cannot be too high. So in order to go through all this, a huge amount of money, you can see $500 million, and huge amount of time, 8 to 10 years is often needed to account for all these properties. And the reason for a lot of drug failure can be the lack of efficiency is around 30%. It can show different adverse side effects. The pharmacokinetics can be a problem. So this is basically pharmacokinetics. Suppose the drug is not getting metabolized, it is accumulating in the body. That is a problem. There can be commercial problems, like the drug is too expensive to be available to common people. Or there can be different other miscellaneous problems which results in the failure of a drug in clinical development. So these pharmacokinetic parameters that we were talking about, which is responsible for 39% of drug failures, what are these parameters? One is the oral bioavailability, that is the fraction of dose that enters the blood circulation after the first metabolism in the liver. Next is the absorption. So how much of the drug is passing into the gut wall? Then is the clearance. So the amount of blood cleared per time unit, and the volume of distribution, that is the dosage divided by the initial plasma concentration. So all these parameters need to be kept in mind, because based on the volume of distribution and clearance, that will give an idea about how often the drug must be taken. If you have to take a drug every half an hour, that is not a very practical thing to do. So that is a very important thing to consider during the development of a drug, that is how often the dosage must be taken. Another thing is the oral bioavailability. So that gives you an idea is how much the dosage must be. So if the fraction of the dose entering the blood circulation is low, that means a huge amount of drug must be consumed for the concentration to be high enough to have any activity. Again, that is not practically feasible. So these pharmacokinetic parameters are very important while developing any drug. And there is something called a penetration rate. So in the site of action of the drug, what is the penetration rate of the drug? So you know that and all of our cells have cell membranes. So the penetration rate is defined by P, that is the partition into the membrane, into A, that is the surface area of the membrane, into the concentration gradient outside and inside the membrane. So that will give the penetration rate, that is how much of the drug is entering into the cells. Now we'll talk about partition coefficient or distribution coefficient D, which is the ratio of concentration of a compound in a mixture of two immiscible solvents in equilibrium. In pharmaceutical sciences, usually both these solvents, one is water and the second is something hydrophobic like 1-octanol. The partition coefficient measures how hydrophilic the substance is, meaning whether it will majorly remain in the water phase, or how hydrophobic the substance is, meaning if it will majorly remain in the hydrophobic phase. So these partition coefficients are useful in estimating the distribution of drugs within the body. Hydrophobic drugs with high octanol water partition coefficients are mainly distributed to hydrophobic areas such as lipid bilayers of cell. Conversely, hydrophilic drugs with low partition coefficients are found in aqueous regions such as blood serum. So depending on where we want the drug to work, the partition coefficient is very important. And so the log of this partition coefficient is defined by log of the ratio of the drug in the hydrophobic solvent divided by the drug in water. And this can, so the drug, suppose it is a protonated drug. So like this acid, some part will exist as the acid and some part will lose the proton and exist in the ionized form. And same in the hydrophobic solvent. So instead of concentration AH, we can write concentration AH plus concentration A minus, because some part of the drug will remain protonated and some will be unprotonated. So this can be rearranged and written as log D is log P minus log of 1 plus 10 to the power pH minus pKa. So the distribution coefficient depends on the pH of the drug as well. Because suppose you have a drug which is highly acidic, so it wants to remain in the ionized form. Then it will prefer the ionic solvent water, which will stabilize the ionized form. Or if you have a drug with very low pH, so it will prefer to remain in the neutral form. Then it might prefer the hydrophobic solvent, which will prefer the neutral form. So the pH is very important for the determination of the distribution coefficient. There is another term called clog P, which we'll look into now. So pH is different in different regions of the body. The intraluminal pH goes from highly acidic in the stomach to around pH 6 in duodenum. And from 6, it slowly increases to 7.4 in the small intestine till it goes to around 7.4 in the terminal ileum. Again, it drops to 5.7 in the cecum and again gradually increases, reaching 6.7 in the rectum. So different regions of the body, you can see different regions shown here. In the stomach, after eating, it is around 3 to 7 and so on. So different regions in the body have different pH values. Now, there is a way of defining solute molecules by identifying isolating carbons. Isolating carbon, or IC, is defined by the carbons that are not doubly or triply bonded to a heteroatom. So in this particular molecule, we will consider this carbon is excluded because it is doubly bonded to a heteroatom oxygen. The carbons on the benzene ring are included. So there are 6 isolating carbon, or 6 IC. And there are other fragments of the molecule, there is a carboxyl group COH, there is a hydroxy group OH, there are electronic interactions, hydrogen bonding interactions. All of these come together to form a term called as clog B. Clog B is a sum of the contribution of the isolating carbon. You can see there are 6 isolating carbon, and of all the different fragments like carboxy, hydroxy, so on. And there are certain corrections made for intramolecular polar, dipolar, hydrogen bonding interactions. You can see there is a certain correction made for hydrogen electronic interactions and so on. All this together give a certain value called clog B. We don't need to understand how mathematically this is coming. You can just remember clog B is a sum of the contribution of the isolating carbon atoms, then the different functional groups like carboxy, hydroxy, and also the different interactions like hydrogen bonding, electronic interactions, and so on. All this is accounted for in a term known as clog P. Now, there are certain rules for intestinal absorption, which are the Lipinski rules. So, any drug must obey all the Lipinski rules. One is the hydrogen bond donor, that is the sum of OH and NH groups, must be less than 5. The second point is the hydrogen bond acceptors, that is the double bonded O, double bonded N, so on. They must be less than 10. The molecular weight must be less than 500 Daltons, and the log P, that is the partition coefficient, must be less than 5. So these are the Lipinski rules for intestinal absorption, and any good drug molecule must follow all these rules. So here are some examples. So this drug called paclitaxel or Taxol, it violates 2 of the Lipinski rules. The molecular weight is greater than 500. It is 837, so it is violating one rule. And the number of hydrogen acceptors, this double bonded O, if you add them all up, then it is 15, which it should be less than 10. So that rule is also violated. So this is not a very good drug because it is violating 2 rules. Now there is a problem that is arising, that is antimicrobial resistance. Basically, humans develop antibiotics to kill bacteria. But bacteria is also created by nature, and so they have a natural mechanism to protect themselves from antibiotics. So as humans, as we develop more and more antibiotics to kill bacteria, bacteria is also evolving to develop resistance for the antibiotics. So now older antibiotics, which earlier used to be able to treat diseases, now they are becoming inactive because the bacteria have developed mechanisms to resist these antibiotics. So that is a problem that is arising more and more as time goes by. You can see that the antibiotic resistance is in 2018 is this red bar for different diseases, and 2050 it is predicted that for several antibiotic resistance, if 2018 if it was 0.1, it is predicted to rise to 10 by 2050. So more and more new drugs need to be synthesized because the old drugs are becoming ineffective. We will now look at a case study involving beta-lactamases. So basically, the hypothesis is that every beta-lactamase has individual properties. So does this mean we have to study each and every beta-lactamase from any organism? That is not possible because there are thousands of genes, thousands of organisms, and so many beta-lactamases cannot be individually studied. So an interdisciplinary approach has been adopted, which combines microbiological screening with computation and experimental techniques. Basically, uh, samples are collected and genes are identified, and so there is homology modeling, and it explains that which genes are the novel mutants. So although different beta lactamases, different organisms have different beta lactamases, that they can be divided into classes based on similarity in their um, uh, gene sequence and so on. So instead of studying each and every beta lactamase, they can be divided into different classes, and computationally also the number of beta lactamases can be narrowed down, and only those which are predicted to be of different classes, they can be expressed and purified in order to design new drugs. So instead of testing each and every beta lactamase, the number of different classes of beta lactamases can be narrowed down by homology modeling of the gene sequences and also from computational databases. So there is this anti-tuberculosis drug, which is developed by inhibiting beta lactamase by a novel boronate transition state analog called BATSA. In 2011, 8.7 million people fell ill with tuberculosis, and new infections happen very fast. Basically, the beta lactamase associated with the tuberculosis bacteria, BLAC, it has identified to be a major resistance factor, meaning that so basically new inhibitors have to be developed, because the old penicillin, you can see, beta lactamase is deactivating the penicillin. So it has developed a antimicrobial resistance towards penicillin. So new inhibitors have to be designed. Basically what happened in the beta lactamase is that you have the penicillin, so cefotaxime has a similar structure to penicillin, where you have a four-membered beta lactam ring. And what was happening is that in the bacteria, the serine was attacking this double bonded. So serine was adding on to this double bonded O. That was the transition state. And the next step was that this was kicking back, and this was, one second, and this bond was breaking. So the four-membered ring was opening up like this, which was deactivating the penicillin. So now the new drug has been designed. So what was happening is that the serine in the bacteria was deactivating drugs which contain the beta lactam ring like penicillin and cefotaxime. It was attacking these drugs and opening this four-membered ring and deactivating these drugs. So the novel inhibitor that was designed was called BATSA, which contained a boronate residue. You can see boron attached to two OH groups, this boronate residue. What this is doing is serine, instead of attacking this four-membered ring, it is attacking the boron, and so it is preventing the opening of the beta lactam ring. So this was the novel inhibitor that was designed, which would prevent the serine from deactivating the beta lactam ring. And several lead compounds were designed containing the boronate residue. You can see BOHOH, the boronate residue is there in several lead compounds. And all of these show different levels of micromolar binding to the tuberculosis bacteria. So these are all possible drug molecules which show decent binding to the tuberculosis bacteria. And all these drug molecules, they have to, so by studying the binding site of the bacterial protein, it was seen that there is an arginine and threonine which binds to a meta-COH group in the phenyl ring. So all these lead molecules, you can see this phenyl ring has a meta-COH group. Same for this, same for this. That is what was observed, is that the meta COH group is essential for high binding. So in that way, the drug was designed, and quite a few good drug candidates were found, in which the most successful was the EC19, which shows like less than 1 micromolar binding, which is quite high binding. So it is a exciting new possibility for an anti-tuberculosis drug. There are some other like case studies also. Basically, there is a molecule called diaza-bicyclooctane. So this molecule, they are a class of serine beta-lactamase inhibitors that use a strained urea moiety. So C double bond O, N is urea. It is strained because it is in a ring compound. So this strained urea moiety is the warhead which reacts with the active serine residue in the beta-lactamase. So you can see here what the boronate was doing. It was taking away the serine. So it was blocking the serine from opening the beta-lactam ring. So that is how the Batsa drug was functioning. Now, in place of the boronate residue, you can have this kind of a residue, which is called a diaza-bicyclooctane. The concept is the same. Here also, the boronate was reacting with the serine and trapping the serine. Here, this strained urea moiety reacts with serine, and this ring opens up. So it is trapping the serine and preventing it from deactivating the beta-lactamase by opening its ring. So this is another case of the serine beta-lactamase inhibitors that uses this kind of DBO moiety. Here also it has been observed that at time 0, where you have the beta-lactamase peak shown in mass spectroscopy, now as more and more of this drug is added, you can see the peak of beta-lactamase here, it is disappearing, because it is now being, this new drug is binding with the beta-lactamase, and this new drug is binding with the receptor on the bacteria and breaking it down. So you see the peak of the beta-lactamase disappears. So this is also another example of a drug that was developed by these methods that we have discussed. So now we will, just 1 second. Now we will look at some practice problems. So the first question is asking, which of the following are processes of traditional drug discovery? So here the answer is all of the above. So if you remember, traditional drug discovery consists of random screening, where from the known medicinal plants or known bioactive compounds, they were randomly screened to find that which of them show activity, or the trial and error method, where different plants are screened to find which of them can act as drugs, or classical pharmacology, which was function based. So suppose a plant was found to have anti-diarrhea properties. So classical pharmacology would go into that plant and look at what are the bioactive compounds responsible for the biological activity it is showing. So all these three are traditional drug discovery methods. There are two other methods. One is the ethnopharmacology approach, where basically the behavior of tribal native groups are studied, who use a lot of ethnic plants to treat diseases, and from there drugs are derived. And another is the serendipity method, which is based completely on accident or luck, like the discovery of penicillin, where it was just left overnight and found that wherever there was penicillin, bacteria was not growing. So these are all the methods of traditional drug design. So the correct answer here is option D, that is all of the above. The next question is asking, which of the following is not involved in rational drug discovery? So here the answer is C, the trial and error method. We saw that trial and error method is part of the traditional drug discovery. Comparatively, the other methods are part of rational drug discovery. Now, rational drug discovery consists of two methods, the structure-based drug design or SBDD and the ligand-based drug design, LBDD. And in structure-based drug design, we know that we need to understand the three-dimensional structure of the receptor binding site. And once we know the three-dimensional structure, we can use computational softwares like docking softwares to predict that what will be a fragment that will bind to that binding site. So that can be done using docking softwares or scoring functions, which will give an idea of the binding affinity of different ligands. Then virtual screening is done. So instead of testing all the possible ligands experimentally, which takes a huge amount of time, effort, money, virtual screening is done where the computer screens to predict the highest binding affinities. So it leaves out low binding affinity and screens and separates only the high binding affinity compounds. So virtual screening is involved in the rational drug discovery. And from there, you have a lead compound, which is then optimized. The other uh, method of rational drug discovery is the ligand-based drug design or LBDD. And LBDD is of two types. Pharmacophore modeling, where you find out that, so from the structure of the active site, you figured out what is the orientation of functional groups needed in the ligand. So suppose we know that the ligand must have two carbonyl groups, one OH group, one aromatic moiety, so on. And then these fragments are linked and joined together. That is pharmacophore modeling. So you can see this is a rational drug discovery method. This is also a rational drug discovery method. Another is the quantitative structured activity relationship or QSAR. So from a list of known binding ligands, a function is derived which takes into account the structural descriptors of the molecule and predicts the biological activity of the molecule. So using QSAR also, we can narrow down the experimental process. We don't need to do experiment for all the ligands. We can use QSAR to predict only the best binding ligands and hence arrive at a lead molecule much faster. So QSAR modeling is also part of the rational drug discovery process. The only option C is not a part of rational drug discovery because trial and error method is a part of traditional drug discovery, which we saw here. So the answer is option C. The next question is asking, what are descriptors in QSAR? So descriptors can be of different types. We know the 0 and 1 dimensional descriptors are basically the molecular weight, molecular formula, number of atoms, so on. The 2-dimensional descriptor is the connectivity. 3-dimensional descriptors are the different atomic XYZ coordinates. And 4-dimensional descriptors are the intermolecular interactions. So what are descriptors in QSAR? Biological macromolecule is not a descriptor. Chemical formula itself is not a descriptor. What is the descriptor is the number of atoms. So 33 carbon, that is a descriptor. The formula itself is not a descriptor. So the better answer is, what are descriptor? Chemical characteristic of a molecule in numerical form. This can be different chemical characteristics like the number of atoms, the different connecting bonds, the 3-dimensional coordinates. So these are all descriptors in numerical form. So it is option A. The next question is asking, atomic branching and ramification fall in which type of descriptor? So the answer here is topological. So basically, topological descriptors tell you all about atomic branching. If you remember, we saw the Wiener's index where so here you have different, imagine this is each of these points are non-hydrogen atoms. So Wiener's index, basically you had to find the shortest distance between different pairs of points and add them all up to get the Wiener's index. And that is a type of topological descriptor. So here also you can see the topological descriptor is telling you about the black atoms, which is bonded to 4 atoms. You can see this black atom is bonded to 1, 2, 3, 4, 4 atoms. The atoms shown with extensions are the face-centered atoms. And atoms outside the cube are shown in white and so on. So this entire description falls under topological descriptors. The physicochemical descriptors are the ones which talk about physical or chemical properties like lipophilicity, hydrophobicity, so on. The quantum chemical descriptors are about the atomic level descriptions in terms of quantum mechanics. And the constitutional or information descriptors are basically how many carbons there are, how many nitrogens there are, that kind of, how many hydrogen bond acceptors, donors, that kind of information. So atomic branching are topological descriptors, that is option B. Question 5 is asking, what are physical properties of compounds? So the answer is all of the above. We know that a compound has a molecular mass. Depending on different molecular mass, it can have different boiling points. Depending on the functional groups, it can have different solubilities. These are all physical properties of compounds, and they will fall. So the boiling point and solubility will fall under the physicochemical descriptors, whereas the atomic mass is also a physicochemical descriptor. So which are the physical properties of compounds? It is all of the above, D. Question 6 is asking, what are the basic requirements for molecular docking? So the answer here is D. So you need, for docking, you need the 3-dimensional structure of the protein, that is the receptor here, and you need a library of small molecules. So either from a database or by de novo designing, you will get a library of small molecules. Once you have the receptor and the library of ligands, the docking software will fit the ligand to the binding pocket and it will predict the binding energy. So the basic requirement for docking is D. You need the protein 3-dimensional structure to get a clear idea of the binding pocket and a library of small molecules to scan to find the best binding ligand. So that is option D. 7th question is asking, which of the following is the docking software? So here the answer is Autodock. Autodock is the docking software that we use most commonly to carry out docking and predict. Docking is basically used for virtual screening of ligands. So instead of testing each ligand individually experimentally, which will involve a lot of time and money, docking software Autodock is used to predict the binding affinities and narrow down the list of ligands to be synthesized and experimentally tested. So this docking software is AutoDock, that is B. So you can see docking software, what it does, it has a library of ligands. It has the three-dimensional structure of the protein. It will narrow down. So it will have a binding energy threshold. All these other ligands are below that threshold. So instead of so many ligands, it has narrowed down the number of ligands to 3, which is matching, which falls within the threshold binding energy. And so instead of synthesizing and experimentally testing all these, now only 3 have to be tested. So it is lessening the experimental time and effort. Question 8 is asking, in virtual screening, hypothetical molecules mean what? So hypothetical molecules basically mean the library of virtual combinatorial molecules. So if you remember, we read a few slides ago, the library of commercially available molecules are the real molecules, and they can be anywhere between 1 to 4 million. And these are normally available with different pharmaceutical companies. The library of virtual combinatorial molecules can go up to 10 to the power 60. And these are hypothetical molecules made by combining different fragments. So if you know different fragments which show binding, these can be combined in different ways hypothetically to have an entire virtual library, which can be used for virtual screening to narrow down the number of possible ligands to be synthesized. So the hypothetical molecules are this virtual combinatorial molecules made by combining different known binding fragments. So the correct answer is option B. Question 9 is asking, what is the docking scenario between receptors and ligands for the lock and key model in docking? So if you remember, the lock and key model, basically you have the receptor and ligand, and both are rigid. So the particular ligand will only fit to the particular receptor. The induced fit model is where the receptor can change its shape to perfectly fit with the ligand, and the ligand can also change its shape to optimize the binding energy between receptor and ligand. So that is the induced fit model. So in the lock and key model, both the receptor and ligand are rigid. That is option A. So the correct answer is option A, where receptor is rigid and ligand is also rigid in the lock and key model in docking. Question 10 is asking, which of the following is present in Lipinski rule of 5? So the correct answer is option B. So Lipinski's rule of 5 is basically the rules for any in vivo drug to be absorbed well and distributed well in the body. These rules are the number of hydrogen bond donors should be less than equal to 5. So hydrogen bond donors are the OH groups, NH groups, so on. They should be less than equal to 5. So this is wrong. The second is molecular weight should be less than equal to 500 Daltons. So this is also wrong. Third is log P, that is the partition coefficient, should be less than equal to 5. So this is also wrong. And finally, the hydrogen bond acceptors, that is the double bonded O. Double bonded O, basically it has lone pairs and it can accept hydrogen. So the hydrogen bond acceptors like double bonded O, double bonded N, so on, they have to be less than equal to 10. So less than 10, this is correct. So the correct answer that which of the following is in Lipinski rule is option B. The next question is asking which of the following approach is considered ligand based drug discovery. So the answer here is both A and B. So uh, rational drug design consists of two kinds of methods, ligand-based drug discovery and structure-based drug discovery. Now, in the ligand-based drug discovery, you have QSAR, that is quantitative structure activity relationship. So this is part of ligand-based drug discovery, where from the known structure of a list of compounds and known function, expression is put together, which predicts the function of any unknown ligand based on its structure. So that is QSAR. Another is pharmacophore, which is also part of ligand-based drug discovery, where based on the binding site of the receptor, the characteristics of the ligand is predicted. So if it predicts that there are two carbonyl groups, one aromatic group oriented in a specific way, that is the pharmacophore modeling, and that is also part of ligand-based drug discovery. Other methods can be by virtual screening of a library of compounds or by searching similarity of known binding ligands. So those are all methods of ligand-based drug design. Molecular docking is not a part of ligand-based drug design. It is a part of structure-based drug design. So in structure-based drug design, you have to know the three-dimensional structure of the protein, so that you have the three-dimensional structure of the protein and the ligand, and the program can predict the best orientation in which the ligand is fitting into the receptor pocket. So docking is not a part of ligand-based drug discovery. It is a part of structure-based drug discovery. So option A and B, that is option D, is correct, which is both A and B. So both pharmacophore and QSAR modeling are considered as ligand-based drug discovery. Now, the advantages of the ligand-based drug discovery is that you don't need to know the protein structure. You can just proceed with the drug design without knowing the protein structure. So the computational cost is much lower, and you have predictive models. The limitation is that since you don't know the receptor conformation, predicting the bioactive conformation becomes difficult. So select, and also another problem is you have to select and optimize the descriptor of QSAR. So QSAR is structure activity relationship. How do you know which structures contribute most to activity? That has to be optimized. Also, the pharmacophore constraints have to be optimized. So without properly knowing the structure of the binding pocket, it is very difficult to design the orientation of the pharmacophore. So that is the limitation. In structure-based drug design also, you have certain advantages because you know the three-dimensional structure of the protein. There is a lot of valuable information about the binding pocket, which helps to design the drug. And it is not biased by any ligand. You can just focus on the target. And because you know the three-dimensional structure of the protein, the bioactive conformation can be easily determined. So, and optimization of ligands is much easier because you know the binding pocket. The limitation is that proteins are flexible. So accounting for that protein flexibility is very difficult. We just saw a few slides ago that predicting the induced fit model is very difficult because the induced fit model, the protein can change its orientation to fit the substrate. So there is a huge number of degrees of freedom, which is difficult for any software to predict. And Other problems are the prediction is done based on molecular docking. So sometimes that can also be like unpredictable, and it is not that accurate sometimes because it is done computationally. So that is about the structure based drug design. What you can just remember is ligand based drug design is of two types, mainly the QSAR and the pharmacophore. And structure based drug design consists of more computational methods like docking, virtual screening, so on. And the final question for today is that which step is not included in de novo drug design? So de novo drug designing, these are the steps. The first step is generating the potential primary constraints, and these are of two types. One is based on the receptor. So the interaction of the receptor forms the basis for the designing the primary constraints of the lead molecule. And the second is ligand based. So depending on the uh, ligand structure, the initial drug molecule is designed. So generation of potential primary constraint is, it is a step in the de novo drug designing. The next step is deriving the interaction site. So finding the atomic level resolution of the binding site to find where there are hydrogen bond donors, acceptors, finding all the interaction sites. The next third step is building up the models. So this is also a part of de novo drug designing. Here you predict certain fragment molecules which will bind with the interaction sites, and the fragment molecules are linked together to design the entire lead molecule. After that, scoring or assay is done to predict the binding affinity of different ligands. And based on that, 5 and 6 are basically about refinement. So refining the lead molecule. In these, so A, B, and D are steps of de novo drug designing, which is not a step. Docking is not a step. Docking is not a step involved in de novo drug designing, because in docking, de novo means starting from scratch, and docking you already have to know the structure of the target and have a library of ligands. So docking is involved when you know the structure of the protein target and the structure of different ligands. So it is not part of de novo drug designing. So the answer of which is not included in de novo drug designing is docking. That is option C. So that is all the slides. And as always, I'll upload this video on YouTube and also share the slides so you can go through them later. I'll end the class now. Thank you.